So I guess we can start.
Okay. Welcome everybody to another Origins seminar. Today we have with us Sukrit Ranjan, who is an assistant professor who just started over at LPL. Sukrit got his bachelor's degree from MIT and then his PhD from Harvard, followed by 2 postdoctoral fellowships at MIT and Northwestern. Sukrit will tell us about his photochemical modeling work and take it away.
Thank you very much for that wonderful introduction, Sarah. So as Sarah mentioned, my name is Sukrit, and today I'm excited to share with you some of my work, which is trying to help us understand life as a planetary phenomenon through the specific lens of photochemistry. So if we step back a little bit and just look at the big picture motivation for this work, really the kind of big picture questions that I'm trying to help contribute to solving are, where did we come from, and are we alone? And these are questions that humanity is engaged with for many millennia now. But our engagement for the longest time was at the level of kind of metaphysics and philosophy. And in the last century or so, we've been this kind of unique and special regime where we finally started to be able to bring empirical tools to bear on each of these questions. So it's really started to move into the realm of kind of concrete science. And this is the opportunity that I'm kind of interested in exploiting a little bit. So life is a planetary phenomenon. Life shapes the planet that it inhabits, and this is our essential paradigm for looking for life elsewhere. In the past 50 or 60 years, we've started scrutinizing the planets within our solar system for evidence of life, and we continue to look there today. We're looking at habitable niches, for example, in the subsurface of Mars, the clouds of Venus, and the interior oceans of the icy moons of the outer solar system. But in the last couple of decades, the field has been revolutionized by the discovery of so-called exoplanets, planets orbiting other stars, which we now know to be extremely common. A number of potentially habitable planets are known. And the question that we have now started to ask is, are any of these potentially inhabited? Is there a way that we can use observational tests to discriminate the presence or absence of biology on such worlds? This is a kind of tall ask. How are we going to scrutinize the atmospheres of exoplanets for signs of life when there are so many light years away? And our essential paradigm for doing so was kind of given empirical grounding back in the early 90s. So on its way to Jupiter, the Galileo spacecraft did a flyby of Earth. When they did this flyby of Earth, a group of scientists led by Carl Sagan performed a thought experiment. They pretended that they were alien scientists and that Galileo was their interstellar space mission. And they asked, purely on the measurements of basis of measurements you could make with Galileo, could you place any constraints on the probability of Earth being an inhabited world? And the answer they came up with was that was a qualified yes, Earth is inhabited. And there was a whole bunch of detailed discussion in the paper, which I really advise you to read. They did a really good job of really pretending they knew nothing about terrestrial biochemistry. But their overall conclusion was based primarily on the existence of oxygen and methane at the same time within the Earth's atmosphere. Those abundances of those gases were hard to explain with known abiotic mechanisms. They then argued that a biological mechanism was somewhat reasonable given that water vapor is present, which implied that the planet was habitable and had liquid water. Although they did include a giant caveat as to whether or not organic life was plausible in an oxygen-dominated atmosphere, which granted but from biochemical first principles, still a bit of still a surprising fact. So that's the essential paradigm for searching for life on exoplanets, is looking for something weird that's hard to explain using known abiotic mechanisms and asking if a biological explanation is reasonable. The basic ingredient for doing this is getting a spectrum of an exoplanet. Today, we are able to measure the spectra of giant exoplanets, planets the size of Saturn and Jupiter. And these days, we're able to do this really exquisitely. For example, shown in this ERS work for the exoplanet WASP 39b, which includes detections of molecules such as carbon dioxide and SO2, I believe for the first time ever. And that's extraordinary that we're able to measure these gases on a planet orbiting so many light years away. So today we're able to measure spectra like these for giant exoplanets. And in the future, we hope to measure these spectra for smaller, rocky, potentially habitable planets. James Webb might be able to do this for the most favorable situated planets, assuming they still have atmospheres. With upcoming facilities like the ELTs and the Habitable Worlds Observatory, due to come online in the 2030s and 2040s, we should be able to do this much more robustly for a much broader sample of worlds as well. And hopefully detect spectra that look something like this, that we might be able to somehow invert and constrain in the presence or absence of life. However, life is also a planetary phenomenon when viewed at the other end. Not only does life shape the planet that it inhabits, the planet shapes life to a significant degree. And in particular, the planet really determines the environmental milieu under which life emerged, and therefore controls the origins of life chemistry that operated to get it started in the first place. With this kind of fundamental insight in mind, so-called prebiotic chemists have done a lot of experimental work to try to figure out how life got started here on Earth. And in the last decade in particular, they've seen a number of remarkable breakthroughs, solutions to really long-standing problems in prebiotic synthesis that have been around for about half a century, including the synthesis of each class of biomolecule from common chemical precursors, the discovery of prebiotic vesicle formation and encapsulation spontaneously under conditions available at deep sea hydrothermal vents, and perhaps most excitingly, the discovery of a pathway for the non-enzymatic synthesis of each of the 4 canonical ribonucleotides, which is something that we've been looking for since literally in the 1960s. So that was a huge deal when that was discovered.
Sorry, so can you go back? Sorry, prebiotic vesicles are what?
Vesicles are just compartments. For modern biology, that's like cell membranes and stuff. Cell membranes are super complicated. So on Earth, what they're looking at there is maybe you have some kind of fatty acid that's present. If you have a fatty acid that's present and you drop it into water under certain conditions, does it spontaneously form a membrane that encapsulates things or not? Or does it remain like as a laminar sheet or like a droplet or something like that?
Okay, so that's what you mean by prebiotic, is basically
Yes.
a fatty acid in water which forms
A
membrane, not a
drop or something like that. Yeah.
And that's a function of, whatever the composition of that acid molecule.
Yeah.
That makes it some surface tension or something. Okay.
That and the pH, which very sensitively controls, does it collapse down like this or does it form? Yeah. There is a problem with all of these chemical approaches, which is that if you diagram out a lot of what the chemistry here looks like, it looks something like this. Your eye is already glazing over. That is a perfectly natural response. The main takeaway from this is that these chemical pathways that people come up with are very complex. There's a bunch of stuff that's going on in them. And this is the main criticism of them, that in order for them to work, they require, yes, they operate from a common set of putatively prebiotically plausible reagents and conditions, but they require those reagents to be brought together in a specific order, in specific pattern, in specific ways. And it's an open question as to how likely that would have been on early Earth without, the presence of a chemist to do it. The analogy that's often used to explain this is a so-called golf analogy, and it's best explained in this quote by Bob Shapiro. So instead of trying to restate it, I'll just read it out. The analogy that comes to mind when talking about these prebiotic chemistries is that of a golfer who, having played a golf ball through an 18-hole course, then assumed that the ball could also play itself around the course in their absence. And this is a reasonable criticism. What it's basically saying is that if you imagine your reaction landscape as a potential surface, if you have a flat potential surface, it's not super likely for your reaction order to go in a specific order from here to here to here to here, as opposed to from here to here or something like that. So that seems not very satisfying. That seems a little bit contingent. How do you actually make that work? And this is a criticism that prebiotic chemists have very much taken to heart. And so what they do now is they look for slow potential surfaces where no matter how you hit the golf ball around, you'll have a preferred outcome, which the system tends to over time. And if we kind of go back to the golf analogy, you can imagine that if you play golf on this golf course over here, if you're a random golfer hitting the ball around, will have the golf ball inevitably go in one direction. That is to say, over the side of the cliff. And so that's what prebiotic chemists are trying to find. They're trying to identify some kind of natural environment where some kind of productive origins of life chemistry inevitably proceeds without the intervention of some kind of cosmic golfer. In addition, in order to make this work, you need the chemistry to be high yield, highly selective. And in a dream ideal world, it would also explain aspects of modern biology. And so this is the work I'm going to be sharing with you today. I'm going to be looking at life as a planetary phenomenon. I'm going to be trying to answer what were the environmental conditions under which life emerged here on Earth? And then what are observational discriminants for planetary scale discriminants for life on other worlds? I'll be covering a lot of ground in this talk, but a kind of linking theme overall will be the importance of the interaction of energetic radiation with molecular systems, or so-called photochemistry. And that's something that'll draw together essentially all the work I'll share with you today. And so with that, I invite you to sit back, relax, and join me on a photochemical journey through planetary environments. I'd like to start on the surface of early Earth about 4 billion years ago, roughly prior to the origin of life. In many ways, early Earth is a completely different planet from modern Earth. It probably had a much shorter rotation period. It was subject to a high flux of impactors, so you had a lot of meteorite strikes coming in. But perhaps the most dramatic way in which it was different is the absence of oxygen from its atmosphere, which is something that was theoretically predicted and then directly affirmed from measurements of the sulfur mass-independent fractionation record. The absence of oxygen means that early Earth must have been a very UV-rich environment. So on modern Earth, we have a whole bunch of oxygen and ozone in the atmosphere, which block the UV from reaching the surface, and that wouldn't have been present on early Earth. And that has a significant implication. Specifically, it means that UV light was probably the most abundant source of chemical free energy to the surface of the planet by a couple of orders of magnitude. So no matter what it's doing, whether for good or for bad, you have to account for the fact that it's probably there. And the fact that UV was present seems to be robust to most of the assumptions we can make about the early environment. It's robust to a wide range of assumptions we can make about atmospheric state, so it's hard to get rid of it in the atmosphere. It turns out that it's also relatively hard to get rid of it in aqueous environments. You can have inorganic anions present that probably block some of the UV, particularly the shortest wavelength stuff, but the longer wavelength stuff, which is where the sun is putting out most of its energy, that still seems to get through. So high UV seems to be something that we have to contend with. And that high UV ends up significantly shaping our thinking, calculations, our understanding of the early Earth in a number of ways. For example, let's think about nitrogen. Where did the nitrogen for the origin of life come from? This is at face value an incredibly stupid question. We are surrounded by nitrogen. It's all around us in the form of N2 gas. The major challenge with this N2 gas is that these nitrogen atoms are joined together by this beautiful triple bond over here. And that triple bond is so strong that it renders those nitrogen atoms essentially inaccessible. It's really hard to figure out how you could get that nitrogen liberated from the gas around us and incorporate it into organic molecules that could then participate in nascent life. The solution to this problem that's been put forward since at least the late 80s is lightning. The idea is you have lightning in an N2CO2 atmosphere that triggers a series of reactions whose end result is the dumping of oxidized nitrogen species onto the surface, which undergo further reactions to give you nitrite and nitrate, so-called NOx. And this is kind of exciting and important because not only is NOx important for metabolism today, but it turns out that in experimental simulations of prebiotic chemistry drives a number of really important processes. It's invoked in processes as diverse as the origin of proto-metabolism at deep sea vents, all the way through to the oligomerization of ribonucleotides. And so that's an exciting picture, and folks worked on this from the atmospheric side for a long time, came up with really detailed predictions that suggested this was abundant in all waters on early Earth. And that's super exciting, but one thing that they hadn't really thought about previously was the potential effect of UV light on the surface of the planet once you have this molecule accumulating in natural waters. This is relevant because it turns out that UV light photolyzes these NOx species, it destroys them. And this is something we know both from measurements in the laboratory and also from measurements in the field. You can go out in ponds and lakes and oceans and things like that and measure the process happening in real time. So nitrate gets photolyzed to nitrite, nitrite gets photolyzed to NO, NO is insoluble and escapes back to the atmosphere. And if you include the effects of this process, it totally changes the answers you get. So that's summarized in this plot over here. In this plot, the x-axis is how much NOx is being generated, is being dumped from the atmosphere to the surface. The y-axis are the concentrations of these NOx species in the early ocean. Note the log scale on both. The gray shaded region is the range of atmospheric NOx production fluxes that are predicted based on models. The dashed lines just demarcate micromolar concentrations, which is a completely fiducial kind of borderline for prebiotic relevance. And the key point to note is that no matter what assumptions you make about the amount of iron that was present or about the about the kinetics of the processes within kind of the uncertainties of the experimental rate constants, you don't really access more than micromolar concentrations of NOx in the early ocean. So that's different from what was predicted earlier for about 3 to 6 orders of magnitude, and that's entirely due to inclusion of the sync due to UV photochemistry. Does this mean that NOx is not available in any prebiotic environment? We argue not. We argue that's not quite the case. We argue that there were some microenvironments in which NOx might have been present at elevated concentrations, specifically shallow closed basin terrestrial waters, things like lakes and ponds that have a large drainage ratio, there you might have accumulated higher concentrations of these compounds because you are able to integrate runoff from a wider geographic area, and you're also diluting your atmospheric column supply over a shallower depths. So in that kind of case, we argue that you could have had significant concentrations of these compounds. And that was a prediction that we made. We kind of, the problem with this is that we kind of worried that you would never really be able to test this in any way because NOx minerals are super soluble. They kind of just, if you run water over them, they tend to dissolve and go away. They don't preserve super well. Fortuitously, geochemists are much smarter and more creative than people like me. And they actually did come up with a way to potentially test some of these. Specifically, Homan et al. 2018 took samples of organic kerogens, that is to say, ancient organic gunk from about 3.2 billion years ago. What was special about their sample is they had samples both from early lakes and from the early oceans, and they identified a nitrogen-carbon isotopic features which were consistent with the presence of elevated NOx in some of the early lakes and ponds, but not all of them, and not in the early oceans, which is broadly speaking consistent with this theoretical picture that I've laid out. So there is some evidence that this isn't just theory land, it might actually be reflective of what's going on in real life. We applied similar reasoning to understanding of sulfur for the origin of life. So again, the basic question we're trying to answer is, on early Earth, what forms of sulfur were available at what abundances and at what environments, with the objective of trying to feedback on some of these laboratory simulations. So obviously you would, in hydrothermal systems, like similar to what you have in Yellowstone, you would have had lots of sulfide, highly reduced sulfur. The question that we asked is, could you have had any sulfite elsewhere? And the reason we focus on sulfite is is when sulfur is injected into the surface of the planet, it definitely on Earth today, and we think on Earth in the past as well, the main form in which it comes out isn't reduced sulfur, it's oxidized sulfur, SO2. And if you have that sulfur in the SO2 in the atmosphere in the absence of oxygen, it doesn't go away quite as quickly. So it could, if you had a liquid body of water in contact with it, it could dissolve into that liquid body of water in accordance with Henry's law, undergo further speciation to give you these S4 species, sulfite and bisulfite. And the question that we would really like to know is, was sulfite available in natural waters on early Earth? And if so, which ones? And the reason we want to know is that it turns out to be really useful in a whole range of prebiotic chemistries. So we set out to build a kind of steady state kinetic model, similar to what we built for nitrogen. The big challenge for doing this with nitrogen is that we have much fewer measurements because sulfite is much less stable in oxic conditions like what we have here compared to the NOx species. And specifically, we found that the range of uncertainties in the literature for the process that was predicted to be the main control on sulfite accumulation, which is disproportionation, was discrepant by more than 2 orders of magnitude. So one study found that this process occurred in a timescale of about 10 days. Other studies established lower bounds of up to 5 years. So those are 2 different numbers. And it turns out that they're sufficiently different that they completely control the calculation. So we set out to kind of try to rule between these 2 contradictory possibilities. To do so, we took solutions of sulfite, we aged them over long timescales, being careful to include oxygen. We used 3 separate techniques to quantify what was going on to that sulfur. Electropotentiometry to directly quantify the sulfite, gravimetry to detect the sulfate, and UV vis tracking to corroborate the whole thing. I'm using the royal we here. Really, I did none of this. All the experimental work was done by my colleagues, particularly led by Khaled Abdelazim. And
Sorry, so thinking about, I know what none of those things mean. Can you explain to me what these tests are?
These are all different ways of figuring out what are the concentrations of different molecules that are present. So electropotentiometry, it's kind of it's a way of directly measuring the concentration of an ion. A pH meter is an electropotentiometer. It's one that's selective for H+. It tells you what is the concentration of H+ and therefore pH. Turns out you can build these for a bunch of different anions. And my colleague Khaled built one of these for sulfite species. So that's a direct probe of how much sulfite you have. Gravimetry, what you do is you take your molecule of interest, you put a salt in there that'll cause your anion of interest to precipitate out. And then you get to remove just that precipitated mass, figure out how many moles of it there is. And then from that, you can back out, okay, how much of that anion was there in the first place? And then UV vis tracking, a lot of these molecules absorb in the UV.
Yeah, that one I could guess based on the, but okay.
Yeah. So, and the key point is we wanted to do all three of these because they had contradictory results in the past. So we wanted to get multiple kind of confirmations within, we wanted to check for internal consistency to see if we could figure out what was going on. Our overall findings were fairly unambiguous. If you look at the amount of sulfite you start with and look at the amount you have left after about 10 months, you find that most of it is left. So that is very strongly consistent with a slow disproportionation of more than a year, and it is highly inconsistent with a fast disproportionation of on the order of 10 days. And this you see both from the electropotentiometry and the gravimetry, and you also get it from the UV vis tracking. There's just very little change in the UV vis over a time scale of almost a year. So naively, this would mean that you have tons of sulfite in natural waters. It's everywhere. You can't get away from it. But there's something I've ignored here again, which is the role of UV. And it turns out that UV light also really efficiently photolyzes aqueous phase S4. And this is both measured in the laboratory, and it's also now used in industry a lot. For example, it's used for detoxification of industrial waste in China these days. If you include the effects of UV, you find that this again suppressed typically sulfite to relatively low concentrations. So here I'm showing a plot similar to what I showed for NOx species. I'm showing the abundance of S4 sulfite. On the x-axis is the volcanism level on early Earth relative to modern day. So this is modern day levels of volcanism. On the y-axis is the concentrations of S4 in the early ocean. The green and yellow curves correspond to the abundance of S4 if you assume inefficient or efficient chemical loss respectively, with the range corresponding to the uncertainty and the physical parameters for the early ocean. And the key thing is, again, sulfite typically does not exceed micromolar concentrations. You could only realistically have the possibility of high marine sulfite if you have very high SO2 outgassing fluxes. This actually has several significant implications. Most importantly, so it means that because SO2, S4 is being efficiently destroyed in early waters, it means that SO2 never saturated in the early oceans. This means that SO2 deposition was always efficient on early Earth, which meant early Earth did not build up a UV attenuating S8 haze. You can then ask the countervailing question, which is that, okay, were there any microenvironments on early Earth where you might have had abundant sulfite? And the answer again is yes. In favorable terrestrial waters, you might have had elevated concentrations of these compounds, either if you had high rates of volcanism on early Earth, as has been suggested from the sulfur mass independent fractionation record, or if you were in, again, shallow UV opaque waters, like closed basin carbonate lakes. There also, you might have had, if you had at least moderate levels of volcanic outgassing, you might have had significant quantities of these molecules. Why do you care about this? It turns out that sulfite is key of several of the advances I showed you at the beginning of the talk. It solves a range of long-lasting problems in prebiotic chemistry. Specifically, if you have sulfite and UV light and carbon dioxide present at the same time, it ends up reducing that carbon dioxide and generating organics in an efficient selective way. Similarly, if you have sulfite and Fe2+ present, it efficiently generates solvated electrons, which can power a range of prebiotic chemistries, including HCN photohomologation. And then for me, again, most excitingly, that sulfite, it turns out to have been really critically invoked in this pathway for the synthesis of the ribonucleotides, which is something that we've been looking to do for about a little bit more than half a century now. You might again ask, this is lovely modeling, but how does this connect to reality? Are there any empirical probes of S4? This is something that we've only started looking at. In particular, I believe Greg Fournier at MIT is looking into figuring out if there's a way to probe if S4 is present on early Earth. But there are already indications that S4 was a prebiotic reagent, and those are coming from phylogenetics. Specifically, people have looked at microbes and have traced back the evolution of those microbes. And they find that sulfur reduction metabolism seems, it seems like the most ancient version of sulfur reducing metabolism is sulfite reducing metabolism. So that seems to be evidence that sulfite at least was common in some natural waters on early Earth. So this is consistent with the consistent but not definitive proof of the scenario that we're laying out. So summarizing the takeaways from this part of the talk, UV was an inevitable part of the early Earth environment, and it had a huge effect shaping the inventories of molecules for prebiotic synthesis. In particular, it restricted most of the atmospherically synthesized molecules to prebiotically significant quantities only in a subset of terrestrial waters and not in marine waters. So this has potentially strong constraining power on some of these origin of life theories. There are tests that are emerging of these theoretical predictions from the rock organ phylogenetics. I'm also excited in the possibility that we might be able to use early Mars as an analogy, as a test case for some of these predictions as well. Overall, our improved understanding of the prebiotic environment is feeding into our understanding of prebiotic chemistry in a synergistic way. So when prebiotic chemists try to do more realistic prebiotic chemistry and with a realistic specific scenario, those chemistries seem to work a lot better. It narrows the parameter space in a useful way for them. So from the surface of early Earth, I now invite you to drift up with me into its atmosphere and into the atmosphere of analogous exoplanets.
Question online.
Sorry, go ahead.
You want to unmute, Andrew?
Yeah, sorry, I was just confused. What is S square bracket 4?
That is a great question. So S4, S square bracket 4, or S sulfur in the +4 oxidation state. So that's what SO2 is, for example. And when SO2 dissolves in liquid water, it makes sulfide and bisulfide. They're coupled by tight isobase equilibria, so like they mutually interchange. So instead of trying to say all three of those all the time, I just refer to them by the broad name of S4.
Okay, yes, as an astronomer, that's confusing. I thought it was perhaps triply ionized sulfur.
Yeah, that is, yeah, I definitely see that. For that, I would have put it as a superscript with a plus or minus up there. But yeah, this is just how reducing or oxidizing is it? Does it like to be connected to oxygens, or does it like to be connected to hydrogens?
Got it, thanks.
So in thinking about early Earth atmospheres, what I'm really thinking about are planets with anoxic CO2 N2 atmospheres. This kind of planet is not a niche case. This kind of atmosphere was characteristic of Mars and Venus, we think, throughout their histories. And it was characteristic of Earth for the first half of its history as well. So in a sense, it's kind of the rule in the solar system. It's based on the solar system, what we think a rocky planet looks like in the absence of life. So it's important to understand both if you want to understand early prebiotic Earth, and also if you want to look for life on other worlds, because it's the biosignature null hypothesis. We think or thought that we understood the atmospheres of these worlds pretty well. In particular, we thought that they were to first order controlled by OH, which is derived from the photolysis of H2O. So OH is extremely reactive molecule. It's affectionately known as the detergent of the atmosphere. So on modern Earth, it's so reactive that it is the main control on the abundances of a wide range of molecules. It also appears to control atmospheric stability. And what I mean by that is, left to its own devices, a pure CO2 atmosphere is unstable. It destabilizes to carbon dioxide, carbon monoxide, and oxygen. But OH saves us from that fate. Specifically, OH, which is ultimately derived from water vapor photolysis, triggers a catalytic loop of reactions whose net effect is to recombine the CO and O back to CO2. So that's why the atmosphere of Mars is CO2 dominated and not abundant, and doesn't have abundant CO and O2. And so this is something that we've understood for a long time from classic work back in the 70s and 80s. And it's something that's well implemented in all of our photochemical models. And it's something we thought we understood pretty well, which is why it was super upsetting in the last decade or so, when all of our models seemed to disagree when we applied them to early Earth-like conditions. That's shown in this plot, in this series of plots over here. This is a little bit busy, so let me walk you through it. And this series of plots, each column is the same planet modeled with the same basic assumptions, but just using two different models. And the thing to note over here is that in this family of models in calculations over here, carbon monoxide reaches percent concentrations purely abiotically for an early Earth-like planet. Whereas in this family of models over here, carbon monoxide reaches concentrations about a factor of 100 less. And this is from classic work now by Sunny Harmon. The problem is even more acute for oxygen, where this model in some cases produces up to 1/10% of O2 purely abiotically, versus this model never produces abundant abiotic O2. And that's again really important for our understanding of O2 as a biosignature. So we did an extensive model intercomparison, and whose goals was to try to figure out what is causing the difference between these models, and we traced it down. The answer was different assumptions regarding the absorption properties of water vapor. This again sounds like something that is first order dumb, in the sense that surely this is something you can measure. Surely it's been measured for a very long time. And the answer is, it turns out no. For a long time, this had very few measurements. Specifically, if you look at the near absorption properties of the water vapor molecule, which is what I'm showing here, wavelength in nanometers in the x-axis, absorption cross-section in centimeters squared per molecule on the y-axis, you find that there was basically no data available longer than about 200 nanometers. So one of the models I showed you earlier, what they did is they incorporated this red line into their models. They took the reliable data that existed up to about 200 nanometers as recommended by the JPL compendium, and that's what they plugged into their model. However, that's not necessarily the most physically correct thing to do. Specifically, it's if you look just at the bond strengths, water vapor should photolize all the way up to 240 nanometers. So what another group did is they put together an extrapolation based on the then current picture of water vapor absorption. They built an extrapolation and they put that into their models. And it turns out that if you harmonize the assumptions regarding water vapor absorption cross-section between models and fix a couple of other errors in each of these models, you can then make them sit up and dance. You can make them agree to each other with it better than a factor of 2. And that's great. So that tells us that we figured out what's controlling this difference. But that's still profoundly unsatisfying because what is the correct absorption across sections? To answer that question, we again did measurements. Here again, I used a royal we. Really, it was my colleague Alexander Fative at the Technical University of Denmark. The data he measured is shown in purple over here. This data has an upward deviation relative to the log linear decay that's the most naive expectation from theory. That is possibly physical. It could be due to something like predissociation effects, but it could also just be that we're approaching the sensitivity limits of our apparatus. And so we don't really trust that. Instead, what we trust is this black line over here, which is a log linear extrapolation of our higher confidence short wavelength data. And the thing to note is that even that lower limit over here is much higher than what was assumed by any model previously. And that ends up having significant photochemical implications. Specifically, if you take the same model, apply it to the same planet, but just vary the H2 absorption cross-sections, it affects the CO concentrations that are predicted by about 3 orders of magnitude. There are also potentially significant effects for the CH4 and H2 concentrations. It removes completely that abiotic oxygen false positive. It suppresses oxygen by 7 orders of magnitude. And it affects the rate at which other molecules are delivered as well. For example, it drastically increases the rate of prebiotic synthesis of formaldehyde, which is also a molecule that's invoked in prebiotic chemistry. So are we done now? Is this problem solved? Regrettably, not completely. It turns out that this kind of problem is fairly common in our models. There's a whole bunch of under-constrained or not fully understood photochemical processes. For example, we recently discovered a new EUV photolysis channel. It has no implications for on the bottoms of habitable planets, but it might be important in their upper atmospheres and in comets. And it turns out that similar uncertainties exist for other molecules like methane, HSO, HNO, and so on and so forth. And we've extended the study for M-dwarf planets as well for oxygen false positives proposed for those. That work isn't completely done yet, but stay tuned. Overall, I feel this points to a need to systematically remediate our models. They're disagreeing a little bit too much. We need to identify the weak points and fix them, and that's what we've started to do. So NASA XRP has generously funded our efforts to kind of do systematic model comparisons, identify kind of weak points and failures in these, or points of differences in these models, and use experiments or ab initio calculations to rectify them, and then feed them back in the models to figure out what the right answer is. So we're working with a really great team, collaborating with folks at UC Riverside, Stanford, and the CFA, and we're really excited to see where we go. Right now, our emphasis is on oxygen and sulfur photochemistry in the TRAPPIST-1 planets, and that's the work that'll be coming out later this year. So summarizing the takeaway from this part of the talk, under constrained photochemical parameters limit our understanding of early Earth-like atmospheres. We've shown that the absorption properties of water vapor, it absorbed more than previously assumed with significant photochemical implications. And it turns out that there's a whole bunch more question marks still to address, and we've started a program to kind of systematically tease those out and remediate them. So from the atmosphere of early Earth, I invite you to drift up with me into the atmosphere of other planets, planets orbiting other stars, potentially habitable ones, and to consider what might be how we might potentially discriminate signs of life on them, whether its presence or its absence. So this is the quest to understand so-called biosignature gases. The granddaddy biosignature gas of them all is O2. O2 has very strongly motivated from the solar system. Again, if we go back and look at the solar system and ask what are the most what is the most detectable ways in which our inhabited Earth is different from the other bodies. It's the presence of O2, which is attributed to oxygenic photosynthesis. And so this means that O2 is a really important and really good gas to look for. There are a couple of reasons, though, that have motivated thinking beyond O2 a little bit. One is the risk of oxygen false positives. It turns out that some of the same features which make O2 such an attractive gas biochemically, that make it such a oxygenic photosynthesis such a productive and efficient process, also make it amenable to false positives, derived primarily from photolysis and escape of hydrogen from water vapor, but also secondarily from photochemical false positives as well. In addition, and to me, possibly the most scary, is the risk that oxygenic photosynthesis is contingent. In particular, there's some evidence, again, primarily from phylogenetics, that oxygenic photosynthesis only evolved once. So it seems that it's hard to make oxygenic photosynthesis go. So there's a possibility that alien biospheres might not evolve if they didn't have that one contingent event trigger. And this has motivated the search for alternate biosignatures. So the idea being that if we discover a biosphere that doesn't, isn't producing lots of oxygen, could we still discriminate it? Could we still identify it? So this has prompted the search for so-called novel biosignature gases. It started with work by Sean Goldman to look at the organosulfurs, and a bunch of other gases have been considered as well. There's a common story you can tell about all of these, though. In general, they have pretty good spectral features. If you see them, you can probably identify them in a telescope. Their known false positive potential is relatively limited, but they're incredibly reactive. They fall apart at the touch of a feather. And it's most likely in most areas of parameter space, in most planetary scenarios, the prospects that they could accumulate to detectable concentrations that you can detect across many light years from a telescope at biochemically plausible production fluxes is very low. It's probably that is the major downside of all of these gases. So does this mean that we're up the creek without a paddle? Not quite. There's a hope, I think, in the form of this phenomenon called photochemical runaway. And I think that can best be explained in this now classic paper by Jim Casting back in 1990. So in this figure over here, Jim is showing the results of a bunch of photochemical models. On the x-axis, you can think of it as the amount of carbon monoxide that's present in the atmosphere. On the y-axis is the flux of CO being injected into the atmosphere, in this case from impacts, but it could really be from anything. And what I want to draw your attention to is this part of the plot over here, where all of a sudden, for very small increases in CO production flux, the CO abundance jacks up by orders of magnitude. And what's going on there is that the main mechanism which limits the abundance of CO in the atmosphere is, again, reactions with OH. The rate at which OH is supplied is finite. It's gated by the rate of H2O photolysis, which is gated both by the H2O abundance, as well as by the supply of photons from the star that are capable of photolyzing that H2O. And so if you inject CO into the atmosphere at rates that exceed this H2O photolysis rate, all of a sudden you saturate that sink, it can build up to high concentrations until it's ultimately limited by surface processes. And this process, we argue that this process is pretty general, and it can occur for a whole bunch of different gases. And if it does, it can have dramatic implications for the detectability of gases that are in runaway. To illustrate this, I'd like to look at the example of ammonia, which has been in this so-called cold Haber world scenario. I don't look at ammonia because it's a particularly good biosignature gas. In fact, it has a number of gigantic weaknesses as a biosignature. I use this solely as a kind of proof of concept, as a kind of case study to illustrate whether or not photochemical runaways are physical, i.e., are they a model artifact or not? And if not, what do they mean for detectability of gases? So this is a plot which summarizes our main results. On the x-axis over here is the NH3 surface production flux, again, log scale. On the y-axis is the column average mixing ratio of NH3. The blue shaded region demarcates the range of pre-industrial NH3 production fluxes. The purple region demarcates the modern NH3 production flux, which is substantially industrial. And just as a kind of extreme upper bound, this green line over here is the net O2 production flux to the biosphere. And the thing that I want to point out is that for a sun-like star, NH3 never undergoes runaway. It's just too efficiently photolized in the atmosphere. It always remains low. You never get this phenomenon happening. But most stars are not sun-like stars. Most stars are M-dwarf stars. M dwarfs are smaller, cooler, they have much lower UV output. And it turns out that if you account for the effects of that lower UV output, that NH3 is able to accumulate to much larger concentrations, specifically right around the upper limits of what the pre-anthropogenic NH3 production flux was, and way before the modern O2 production flux. This NH3 can undergo runaway and jack up by orders of magnitude in concentration. And this can have really significant implications for the detectability of NH3, and that's what's shown in this plot over here. Here I'm showing a simulated JWST observation of the so-called cold Haber world scenario. The orange and green lines, the green line is a model as a planet with no NH3 production. The orange line is an atmosphere with just pre-NH3 production, pre-runaway NH3 production flux, and they're indistinguishable. NH3 is basically invisible. The blue line, by contrast, is a planet with an NH3 production flux just twice that of the orange line. But because it's at that runaway, it goes into that runaway threshold. All of a sudden, it floods the atmosphere, it reaches the upper atmosphere into the stratosphere, which is accessible to transmission spectroscopy, and all of a sudden it's super detectable. You could detect NH3 at these abundances in just a few transits with James Webb. And if we go back and do that analysis for all these other gases as well, we find that these gases, which are invisible in all cases at biotically plausible production fluxes for planets orbiting sun-like stars, are for low-mass M dwarfs in particular all of a sudden detectable if they're able to access this runaway regime. And this has significant implications. It means that planets orbiting M dwarf stars, if they can retain their habitability, if they can retain their water in their atmospheres, are uniquely accessible to biosignature search, and therefore in testing theories of prebiotic chemistry. So as we discussed earlier, M dwarf stars have less UV than sun-like stars just because of their cooler photospheres, and specifically at the near UV wavelengths that actually make it to the surface of early planets. And this is important because from a habitability perspective, the problem is not that M dwarf UV is too high. If you calculate what the UV environment is on an M dwarf on a surface on an M dwarf planet, if it maintains an atmosphere, it never even has as much UV as early Earth did, just because it has less near UV emission to start with. So the problem with M dwarf planets is not that they have, assuming they maintain their atmospheres, is not that they have too much UV. It's that they may potentially have too little UV. This might sound, again, like, I think this is a third thing I've said that sounds incredibly dumb. How can lack of UV ever be a problem? UV is terrible, it gives you skin cancer, less UV good. But it turns out that while UV is definitely bad for life once it's established, there's some lines of evidence that suggest that UV may have been important to get life started in the first place. The first, and to me most compelling, is kind of experimental. If you just try to make molecules in the lab, it turns out that the most productive pathways typically are the ones, many successful pathways, in particular, many of the ones that I showed you earlier, fundamentally are gated by the amount of UV they get. They don't work in the absence of UV. The second, again, is theoretical. UV was not only an abundant source of chemical free energy and earlier, that it's capable of affecting irreversible changes in the molecular states of in the electronic configurations of molecular systems. So it can actually generate localized changes in entropy, which thermal sources of energy can't do. And then finally, and for me, there's a historical line of evidence, which is that all the molecules, the biogenic nucleobases, the informational components of the genetic code, all seem to be really UV photostable. So it didn't have to be that way. So here I'm plotting how UV resistant different nucleobases are. And the nucleobases that life uses, which are these blue ones, are all extremely UV photoresistant. Whereas other nucleobases that life could have used but didn't are all extremely vulnerable to UV attack. And so that's interpreted as evidence that the nucleobases emerged, at least emerged in a high UV environment. And so this has given rise to the question, is UV required to start life? And this is something that Paul Rimmer at the University of Cambridge has been looking at very heavily. Specifically, he's been looking at ribonucleotide synthesis through HCN photohomologation chemistry, which requires UV light to succeed. And the specific conclusion that he's come up with is that UV light probably is that the lowest massed M dwarfs, and the lowest massed and most inactive M dwarfs, don't emit enough UV light to get life started in this way. And this is something that we've been looking at as well. We've been getting new measurements of UV emission of young M dwarf stars with new facilities like AstroSat. And broadly speaking, we agree with this conclusion as well. So does this mean that these kinds of origins of life chemistry don't work for M-dwarf stars? We can't say that yet because there's still a big caveat about flares. M-dwarfs are flare stars, they flare frequently, they flare often. And it's possible that intense but transient flare UV might substitute for low steady state UV. And Paul is doing experiments there as well. I really hope the answer he comes up with is no, because if so, then it means that life search on exoplanets has the potential to test theories of abiogenesis. Specifically, if you find that some of these M-dwarf planets don't have enough UV to get life started in the first place, but you detect life there anyway, it tells you that the origin of life does not require UV light, which to me would be a really exciting thing to figure out. So there's still a bunch of question marks there. Specifically, we need to understand the UV evolution of near UV emission of these stars as a function of their ages, and that's work that we're engaged in now. So to summarize takeaways from this part of the talk, the perfect biosignature does not yet exist. The major problem with the so-called unconventional biosignatures is that they're exquisitely reactive and unlikely to build up to detectable concentrations. The one kind of avenue for hope for these reactive gases is that they're particularly detectable in the near term via this phenomenon photochemical runaway. And that's something that James will be able to see, and we've already started scrutinizing those planets. One exciting idea for me is the idea that we might be able to use these M-dwarf planets in particular as an opportunity to test theories of the origin of life as a way of falsifying some of them. We still need to do a lot of observational and theoretical experimental work to figure that out, but we're engaged in it now, and I'm excited to see where it goes. So just to summarize the talk overall, in terms of big pictures, UV photochemistry is key to understanding life as a planetary phenomenon. It controls the milieu under which life emerged here on Earth, and therefore is a key input into our laboratory simulations of origins of life chemistry. It controls the atmospheric discriminants with which we hope to differentiate living and dead worlds. And perhaps most excitingly, through its important role in origin of life theories, it offers the potential to use exoplanet life search as a way to test theories of abiogenesis. We still have a lot of theoretical, observational, and laboratory work to do to realize this promise, but we're engaged, but it's underway. And I'm really excited to be working on this with a bunch of really great colleagues and collaborators. And yeah, I'd love to take questions. Thank you.
So maybe you said this and I missed it, but can you speak a little bit about the relevance of M-dwarf flares in this constraint? Because I guess I often think of them as being at some point in time very UV active.
Yes. So there's two things that we have to think about. The first and most fundamental thing is if you have a lot of UV for a short period of time, does it work that is the production of these biomolecules just linear, like you just produce a whole bunch of it and then they stick around for a while? Or do you trigger fundamentally different excitation, fundamentally different modes? For example, if you produce enough radicals, you all of a sudden trigger strictly destructive chemistry instead of the somewhat constructive chemistry that's been observed so far. So that's a purely experimental chemical question, and it just hasn't been answered yet. I know that some teams are starting to work on it, but we don't have an answer for that yet. The second kind of question that you're getting at is, well, okay, let's suppose that that does work. So let's suppose that it works chemically. Early on in these M-dwarf histories, we know that they're very active. They have both higher steady state emission and then also higher flare rates. So in that case, isn't this problem just solved? If it works chemically, shouldn't it just always work? The answer to that is also not necessarily, we're still not completely confident of the answer there, and that's because of two things. The most important of them, in my view, is that we still don't know if the planets that are habitable for evolved M-dwarfs were habitable at the beginning. And that's because the habitable zones drift over time. So we know that planets that are habitable now were probably interior to the habitable zone at the very beginning. And so the question is, was there ever a period where elevated flare rates and UV output overlap with habitable conditions for planets which are today habitable? And that's the question that we still haven't answered yet, but that's the question that stellar observations and stellar theory will be able to answer for us, I think.
So you're saying, yeah, because I guess I'm questioning whether or not the detection or lack thereof of biosignatures on an M-dwarf really tells you anything about the need for UV. So how would you go about constraining it? So what is it that stellar evolution models or stellar or observations are going to tell you? How does that, I don't understand.
So this is work that Evgenia Shkolnik and Travis Barman have started to do, and their collaborators and colleagues. They've been able to do it in a kind of very broad error bar population mean sense. What you really need to understand is what is the precise evolution of the total amount of UV output of the star as a function of time? How does that compare to the visible and near IR output, which controls the climate? And in a coupled sense, was there ever a period of time where large amounts of UV, of near UV coexisted with the right range of visible and near IR to sustain globally climate conditions. Because right now, my understanding is that if you just take the middles of those distributions and extrapolate them outwards, the planets which had enough UV would also have been in a runaway greenhouse state and would have been uninhabitable at the time. So like that's the big question there. Did you ever have a habitable non-runaway greenhouse state coexisting with lots of UV?
These flares are like orders of magnitude different, right? So I guess if you could do it with a flare, how could there not be overlap between those two populations?
If you can do it with a flare, then there is a category of objects, of evolved objects which fall into that criterion. So we did a little bit of work on that. It was led by Max Gunter. Other groups have started doing that as well, looking at test flare statistics. So it doesn't work for all M dwarfs. It does work for a subset of them. But like, there's still a bunch of like big. There was a lot of assumptions that were fed into that work, which we knew. It was a first step, and we're working on refining them now and getting a better understanding. So like, ultimately, I'm optimistic that this will be a probe that we can use, assuming the chemistry works out. And so that's one of the reasons I'm super excited about the potential life search on planets orbiting M dwarfs.
Kind of related to that, before we go online, you've talked about flares in the context of biosignatures, and then you showed it very briefly. How does that relate to photochemical runaway? Like, is a flare enough to overcome that threshold we need to prevent runaway from happening?
That's a great question. It totally might be. And that's not something I looked into. We assumed a relatively inactive M dwarf spectrum for that reason.
So open question.
Yeah, that's an open question. We have not looked at the transient time dependent effects of this phenomenon on runaway or anything like that. People that could do that really well, in my opinion, are probably Antigona Segura and Howard Chen.
Andrew?
Yeah, thanks for the talk, Sukrit. So I just wanted to check if I understood the arguments that the UV is important for biogenesis. I guess you showed some data that the DNA has has evolved to be, relatively impervious to UV. I guess I'm sort of wondering if that's, I guess, I don't know if I quite understand the full logic behind that argument. It's like, well, it may be, evolved that, durability to UV because it needed to, but life somewhere that didn't need to evolve that might not.
That's absolutely correct. So there's a possibility that there might be multiple paths to the origin of life. Even if life went through a UV experience of UV-rich evolution here on Earth, maybe it didn't elsewhere. So that's entirely something that's possible. That falls into the category of things occurring differently from other planets as they did on Earth. So we definitely have to be open to that. So that's why if you detect life on one of these low UV M dwarf planets, it doesn't necessarily mean that life must go through. Life does not have to go through UV. It might be possible to have a UV dependent origin of life as well. All it tells you that UV is not required for the emergence of life, which is a proposal that some folks have made. So it's a way of falsifying something in a field where it's hard to get falsifiable constraints.
And yeah, sorry, this is just my basic understanding. I don't know if you said this, but one of the arguments for UV being important is just that, it's sort of the same reason that it's harmful, right? That it's just you need mutations for things to happen. And then obviously once something works, you don't want those mutations, but until you have something that works, you do.
Yes, that's correct. Yeah, it's chemical free energy. And chemical free energy is super useful right up into the point where you develop enzymes, and you can start harvesting it from the environment in much safer and more stable ways.
Thanks a lot. Thanks again. Yeah.
Other questions for Sukrit?
Just a super simple question. So when you were discussing the photochemical runaway, so the models you were showing where the abundance increased by a lot in the atmosphere, were those still using those were still surface fluxes consistent with biotic outputs, and that was the only change was the M dwarf star versus a solar star? Okay.
That's correct. Yeah, we kept everything the same. That's the only thing we changed. We kept the climate the same. We kept everything else the same.
Oh, cool. Thanks.
Any other questions? If not, Sukrit is around, so you could find him and chat more, but otherwise, let's thank him again for a great talk. Let's start recording.